Hello and welcome to our tutorial on building custom MASST databases. MASST is a cutting-edge algorithm for searching mass spectrometry data. Similar to the BLAST tool in genetics, MASST searches entire databases for matches to an input spectrum. And because MASST can search billions of spectra in seconds, this tool enables you to answer the question, "Where have I seen this molecule before?" in about the same time it takes to ask it. MASST results can also be paired with metadata to track the occurrence of a molecule across species, strains, or even product types. These metadata-rich databases are often called domain MASTs. Ometa Flow includes several public domain MASTs covering hundreds of thousands of samples from microbes, plants, tissues, foods, etc. However, we also allow you to create your own MAST databases tailored specifically to your applications. Today, we're going to walk through how to create custom MAST databases and visualize your results. So here we're creating a database of water samples for tracking molecules across river, sea, and drinking water in different areas. Here's a look at the metadata file with information on each of the samples. The only required column here is the file name column with the names of the samples you're adding. For the other columns, you can add metadata on its own or as part of a hierarchical tree. For example, here the collection IDs are on their own, while the columns tree 1, tree 2, and tree 3 contain hierarchical data. In this case, the first tree level separates samples into different collection areas, the second into different water types, and the third into year of collection. You'll notice I didn't include that last level for every sample, which is totally fine. Now there are two options to add tree information to your metadata. The first is exactly what's shown here, with columns labeled tree 1, tree 2, tree 3, etc. for the different levels of the hierarchy. However, if you already have column names you like and don't want to change them, you can use the second option. For example, here we have data from different batches of salmon processed through different factories at different times of year. Instead of renaming the factory, season, and lot columns, we can keep those column names and upload an additional hierarchy file. This file lists the names of each column that contains tree information and their level in the hierarchy. Once you've finalized your metadata, you're ready to build a database. To do so, go to the MASST build workflow, which you can access through the Workflows tab here. In the workflow input, you can enter a job description and then select the mass spectrometry files you want to add to the database. Next, you'll choose whether the database is public or private. A public database can be added to or updated by anyone within your organization, while a private database can only be updated by your user. Both public and private databases will be available to search by anyone within your organization, but will not be accessible to anyone outside your organization. Next, you'll select that metadata file you created and enter the name of your MASST database. If you're adding new files to an existing database, you can enter the name of that database here, otherwise just add a new name. Finally, you can add some optional information about the samples you're uploading. It's not necessary, but can be useful if, for example, you know all the data was collected on the same instrument. Once you click Submit Workflow, the MASST database will start building. Once it's finished, you'll see the new database as an option to search in Ometa MASST. For example, let's say I find this molecule in a molecular network. It doesn't have a library match, so I don't know exactly what it is, but I can still find out where I've seen it before using MASST. To do so, I'll click the MASST Spectrum button here. I'll choose my new water database and click MASST Molecule with USI. In a couple seconds, I'll get my results. I can get information on matching datasets, files, and individual matches, and visualize that hierarchical tree using the Custom MASST tab here. Here we can see that this particular molecule was only found in river water that was collected in year 2, but was found across both of our areas of collection. Also, just for fun, here's what a search using that salmon database looks like. This molecule was only found in Factory A, mostly during the summer, and exists in lots 1, 2, and 3. Custom MASST databases can be built from public or private mass spectrometry data with as many samples and whatever metadata you can think of. But for now, that's the end of our tutorial on building custom MASST databases. Thanks for watching.